Hello again, and welcome to the 2013 Hutchinson Award presentation. It's my very great pleasure to introduce this year's winner, Curtis Suttle. I think one of the marks of true insight is to take something that's obscure and make it obvious. And I think that's what Curtis has done all the way through his career. Now we take it for granted that viruses are important in biogeochemical cycling, that they fundamentally alter the ecology of phytoplankton, that they interact with microbial consortia in ways that are obvious, and even something that we can look at them and count them with light microscope or epifluorescent microscope, and you'd think that doesn't make any sense. And it didn't maybe 10 or 15 years ago until Curtis got involved. He's been cited as one of the world's leading marine virologists, and I don't think there can be any doubt that that's true. His research spans so many different areas that it just feels a little bit like a laundry list to go through them, but I'm going to do a little bit of that anyway for particularly freshwater scientists who may be less familiar with his work. He's one of the very first scientists to look at the role of viruses as pathogenic organisms to phytoplankton. He's been instrumental in categorizing and quantifying the biodiversity of viruses, particularly in marine systems and extreme environments, as well as taking techniques out of other disciplines and applying them to the ecology of viral particles. This work has revealed entirely new families of viruses that were not known before. They've increased our understanding of the interaction between the physical environment, such as UV radiation, and viral communities. And as I mentioned, the one that I'm shocked at was the idea that I could actually use a microscope to count viruses. And I'm still trying to convince Curtis to work in inland waters, saline lakes, lots of viruses. At any rate, Curtis is amazing. He does it all. He's served on the ASLO board. He's on several editorial committees and published well over 100 papers. His work's often in the very best literature, in the very best journals, ours as well as others. And really, it's one of those things that when you sit on a committee, you just look and go, how can I actually believe that anybody can be this good? So anyway, with that, I'd just like to introduce Curtis and his presentation for today, which is the G. Evelyn Hutchinson Award for Excellence in Limnology and Oceanography.
Thanks, Peter. Thanks very much. Thanks. Thank you.
It's childproof. I can open it. It's a nano, isn't it?
How do you open it?
It's locked.
It's locked. There we go.
There it is.
Thanks very much. Thank you. That's great. Thanks very much. Good evening. Thank you. So this is probably the toughest crowd to talk after the business meeting crowd, so as opposed to the after dinner crowd. But I think for me, it was just amazing. ASLO's the very first society that I gave my first talk at an international meeting, and it's wonderful to hear and to see how vibrant it is, how it continues. Sometimes I think against all odds, but it continues to get better and better and better and bigger, and I think serves us in more diverse ways than it ever has. So all the way from publishing the best journals, I think, in the field to embarking on areas of policy and public outreach and all kinds of things that we never did 10 or 15 years ago. So I'm very proud to be a member, and I couldn't be any more proud than to receive this award. I'm truly honored. When I look at the folks that have received this award before, I consider it, I would have never believed that I would have been on that list of individuals that have received this award in the past. And so I thank, of course, the people that nominated me, which is a selfless task that requires a lot of effort. I thank the committee for doing it, and of course I thank all of those people that I've worked with over the years who ultimately should be sharing this award. But today I I get to talk at the end, I understand, because, you know, before I only had 30 minutes, now I've got 90, right? So no, well, I'll make sure I get you out of here in time for dinner. And it's going to be a short talk, but I would say that this is probably one of the most difficult talks I've had to give, in a sense, because it's not a 15-minute scientific talk, it's some blend of where you've come from, where you are, and where you're going to. And the one thing I thought, this is supposed to be a mid-career award, so the only worrisome thing, I guess I'm going to have to be steaming along when I'm 90, right? So, okay. So what I'm going to talk to you about today is basically I'm going to take you a very brief voyage to the Virosphere. And the Virosphere, if you haven't thought about that before, is in essence having a little, there we go. So it's all those areas of the Earth where viruses exist and ultimately are affected by viruses. And what's really changed in the last 20 years, we know that that's basically where all life is, and even in areas where our definitions of life don't exist. So for example, when I talk to astrobiologists, I say they should be looking for viruses, because viruses are the easiest things to find. They aerosolize there, and they're at order of magnitude higher concentrations than any other kind of microbial life. So what I think about, and my group thinks about, I think about viruses. So we do a lot of work on viruses, individual viruses. I think about their diversity and their role in their evolutionary history and what they do in the environment. And so how do we find our way as scientists? And I'm no more intelligent than anyone else, and my insights aren't necessarily deeper than anyone else. But if you kind of look at this as individuals together, and each one of these circles are all the way that we're influenced in the decisions that we make as we move along as scientists or in our careers, and we think each one of those circles, one could be mentors, one could be funding, one could be where you went to school, one could be your opportunities, and so all of these things come together, and for each one of us there's kind of a little sweet spot where we end up where we are. And so what are all of those things that kind of merge us together? And I wanted to spend 5 minutes maybe talking about at least my perspective and where I think is relevant to kinds of things that I've heard over the years and certainly have been influential in the way that certainly I've thought about science. And so the one thing, I'm a little bit unusual because there's not many kids that had sailed around the world on a sailboat before their 15th birthday. And so you would say, well, that's kind of an unusual upbringing. That doesn't make a, you know, I can't really relate to that. But the thing that I remember more than anything else is when I was about 12 years old, we were on an island in the Great Barrier Reef. And we happened to be tied up at this island for about 2 or 3 days. And the Alpha Helix was there. And the Alpha Helix was a ship, an oceanographic ship. I think it's run by maybe the University of Alaska now, but it was Scripps Institute of Oceanography. And I think this was its maiden voyage, that's how long ago it was, to do expositions on the Great Barrier Reef. And there were scientists all over that were doing things. And I was a 12-year-old boy running up and down the beach talking to all of these scientists. And it was honestly at that very moment I can look back to that and say, this is what I want to be when I grow up. And so my point here is don't underestimate, for all of us, for educators, for parents, as mentors of students, don't underestimate the influence you have on other people's lives. And there seems to be that sweet spot I've heard over and over again. Kids sort of between 10 and 15 years of age, but even younger, will suddenly have an epiphany or something that they want to do, and yet you will never know that you were the catalyst that had that effect. And so that's both negative and it be positive. If I look back at my life, those people that had positive effects, those people that had negative effects, in most cases they had absolutely no idea. So the point is that think about it when you're interacting with individuals, with young kids all the time, probably in different things, you can have an enormous impact on their lives without ever knowing it. So catch them when they're young. The other thing I was really fortunate to as an undergraduate, I just managed to hang around with the most incredible group of ecologists that I think anybody could hang around with. And they were so critical. They just hammered you as an undergraduate student. They questioned everything you said. They criticized. They say, why do you say that? How do you interpret that? Read this paper. And so I wish I could be as good as they were at teaching people to think critically. And so I try and do that. I don't know if I'm as good as that, but I think that that's so important to tell our students that are there to not only think critically, but ourselves be critical. Not in a negative way, but make sure that you think about what you read, because there's lots of things out there that are maybe not quite what they've said. Those data aren't quite interpreted correctly, but I think that's another thing. So when we get to that era when people are transitioning and now they're in universities, if we're going to create good scientists, we have to teach them to think critically, and we have to do that ourselves so that we can be a role model. And finally, I'd say take a chance and go after what you want. And so I guess this is the story of my life. I wasn't a sterling student, but I knew, you know, here I was as a beginning graduate student a few years, you know, quite a few years ago. And I knew who I wanted to work with, who I wanted my PhD advisor to be. And I thought there was absolutely no way in the world that he would accept me. And he wouldn't. So I just kept hammering on him. I hammered on him for a year because I knew that I wanted to work with this fellow, Paul Harrison, who is a physiological ecologist at UBC. And I just loved the way he said, the way we do science is we critically evaluate things in the lab, we take it out to the field, and we test those ideas in the field, and we see if they make sense, and then we make more hypotheses in the lab, and we test them in the field. So 1 foot in the lab and 1 foot in the field. And I can see that that's all Paul Harrison's students came out like that, and he's a wonderful guy and a wonderful mentor. But I just said, if I'm going to work with this guy. And so always, if they take a chance and go after what you want. And this is also true of my partner in life, Amy. So I was a graduate student here, and I said, this wonderful woman, she's bright, attractive, intelligent in all sorts of ways. And I would say she's been probably has the biggest impact on my scientific career, and we've been it's been like a family business for many years. And so again, did it seem possible at the time? No. If I told you why it didn't seem possible, you'd be astounded. But together, it took many years, and ultimately that's something else I reached for. And then the final person here is Jed Furman, and he may not even know it, but I had picked him out to be my postdoctoral advisor while I was still a PhD student, and I didn't really know how that was going to happen. But I had seen, you know, Paul Harrison and the tradition of Dick Dugdale and all these wonderful scientists, and I said, well, where is the field moving? And I said, it's microbial. And I looked at Jed Furman, a young scientist coming out of the tradition of Larry Pomeroy and Farouk Azam, and just kind of opening those windows of microbial ecology in the ocean. And I was, I guess, at that age where, you know, people really still didn't think that microbes were all that important. And, well, microbes, they didn't consider phytoplankton microbes. Those were phytoplankton, right? And so all of these kinds of changes. And so, again, I was lucky enough to have that opportunity to work with Jed. And that turned out to be incredibly important because while I was working in Jed's lab, he had a PhD student called Lita Proctor. And although I had gone to Jed's lab to kind of test, my idea at least was to test some of the ideas and merge it, some of the ideas I got from my PhD thesis and try and merge it with the kinds of things that he was thinking about, I saw Lita there, and she was working on all of these, like developing these methods and looking at viruses, and all of a sudden there was this whole enormous universe that really nobody knew about. And interesting enough, I thought it's really appropriate because it was 25 years ago, in 1988, I realized 25 years ago that Lita gave the first talk, at least that I'm aware of, talking about the huge concentrations of viruses in the ocean. And so if I hadn't made that connection, then probably I wouldn't be working on viruses in the sea, right? And so then at that point I took a job at University of Texas, and what are you going to do as an assistant professor? And I thought, well, let's start looking at viruses infecting phytoplankton. And my final thing in terms of things to do is try and surround yourself with people that are great. And I don't know how I was so lucky. You can see me there, I look like I'm about 19. But, you know, so my first crop of people that came to work with me, and I realize now that they were kind of doing the same thing that I did. I had Steve Wilhelm and Marcus Weinbauer as my first postdocs, and Steve Short and Matt Cottrell and Feng Chen as my first PhD students. And you know, here I am, an assistant professor, and I realized that these folks were doing the same thing that I had. They were trying to identify areas that they thought, and I can remember them contacting me saying that they would want to work in this area. And so, I mean, I was incredibly unfortunate, and if it wasn't for those folks, and Amy that you see there in the lab as well, if we hadn't all been working as a team, I'm sure I wouldn't have been as successful in my career as I've been. So I think to the extent that you can, to the extent that you can control it, surround yourself by excellent people and become involved with excellent people. Right? So it goes both ways. If you can create an environment in which people are attracted to, that's great. And you can also join people that are excellent as well. And so I think that's tremendous. And so if we go back then, the first paper that came out was shortly after Lita gave her talk, was by Gunnar Bratback's group. And interesting enough, they got the Martin Award for the whatever it is, the most influential paper, I think it was last year, and about high abundances of viruses in aquatic environments. And it was those observations that just changed the way we thought. And then Lita and Jed's paper came out shortly after that, again, not only documenting the high abundances, but the role that they were having in microbial mortality. And then a little bit after that, we had a paper that came out talking about the role of viruses and infection of phytoplankton. And so that was those three papers that kind of really opened up the field a long time ago, in 1990. And so I can't give a talk without showing this slide, so I apologize for all of you that have seen it before. But so we know that there's lots of viruses in the ocean. And so I did this calculation one day, and I strung thought, well, what if we took all the viruses in the ocean and we strung them end to end? How far would they go? And so it was easy to calculate the number of viruses in the ocean. We just took the minimum estimate that there was anywhere, about 1 million per mil or so, and I assumed that they're about 100 nanometers long. And so we have 10 to the 30th viruses in the ocean, about 100 nanometers length. So they would go 10 to the 23 meters if you stretch them end to end. That's a big number. That's 10 to the 20th kilometers. That's still a huge... How big is that? Well, let's just divide by light years. So they would actually go 10 to the 7th light years if you stretch them end to end. So that's a lot of virus particles. So that's, the nearest star is Proxima Centauri at 4.2 light years. The Crab supernova is 4,000 light years. The Milky Way is 150,000 light years across. So in fact, those viruses would stretch further than the nearest 60 galaxies. And that might seem like a trivial calculation, but you have to realize that every one of those viral particles contains its own genetic information, and that those viruses, based on the calculations that we do, are responsible for about Avogadro's number of infections per second in the ocean. About 10 to the 23rd infections. And so they're incredible. Every time they have an infection, there's an opportunity to laterally transfer DNA. They're having big impacts on the way processes are working in the ocean. And the amazing thing is that 25 years ago, no one had any idea at all. And that's one of the really exciting things about the field that we're all working in. There's lots and lots of things to discover. So if we think about it, so why does anybody care? So we think about 95 to 98% of the biomass in the oceans is microbial right now by weight. Those microbes produce half the oxygen in the earth, and our estimates, our best estimates right now, are that viruses lyse about 20% of that biomass every day. So when they really turn things over, so as a consequence that they really catalyze the movement of nutrients from particulate matter back into particulate and dissolved organic matter. So this is what Steve Wilhelm and I turned the viral shunt quite a number of years ago. And so ultimately what we know is that they release that organic material, they stimulate respiration. Ultimately what they're doing is they're changing the pathways of nutrient to energy cycling in the ocean. It's always been there, but it's just one of those things that we didn't account for. And so if you think about it, one of the things, here's Marcus Weinbauer and Steve Wilhelm again, and you might wonder why they're smiling so much. Well, the reason that they're smiling so much is it took 20 years to publish this paper. And so we started this work when Marcus and Steve were postdocs in my lab, and by the time we published it, Steve is a full professor and Marcus Weinbauer is a senior scientist at the CNRS, right? So, and the reason it took so long is we didn't really understand what was happening, because we did experiments, and what happened is that when we removed the virus's primary productivity stopped. And this is not what we were thinking at all. We were thinking that viruses would be actually increasing the mortality rate of phytoplankton. But in fact, it was exactly the opposite. And so when we first did these experiments in Texas, I didn't have an explanation for it, so I wouldn't publish it. But we repeated them so many times, and then I was on sabbatical in Villefranche. Amy and I were there, and we did more experiments. And ultimately, they were repeatable, and then our colleagues in other areas were saying they were seeing the same thing. And so it was clearly happening. And so what we see in this particular graph here is that we've got growth rate of Synechococcus, which is one of the major primary producers in the ocean, on 3 different experiments. And essentially what we've done is we've diluted the viruses out, and we've we've concentrated them. Half of them we heat inactivated by gentle heating, and the other half we just added back at the same concentration. And that you can see here is that if we look at the growth rate, when we add the viruses up here, growth rates are much higher than when we remove the viruses. And so that was something, again, that kind of changed our way about the way nutrients and energy are cycled. And then if we go back and we start looking at some of these other things, Emma Shelford, who's a PhD student in my lab, and we were collaborating with Matthias Middelboe, we started looking at what's the mechanism of this. And so what we see here is an example in which we have chlorophyll and algae from 3 different locations. And again, with viruses, we see that over 24 hours, this is change in chlorophyll. So this is either negative or positive. And so we see when we add viruses, both in a productive coastal environment or even in the very oligotrophic Indian Ocean, that we see changes, significant changes in the amount of chlorophyll that's there. And ultimately, what that's correlated to then is the amount of ammonium that's produced. And so it turns out that viruses are really intimately involved in the recycling of nitrogen in this system. And so we can see here that when we add viruses, so those are the ammonium concentration, and the top is a productive coastal area, and the bottom is the oligotrophic Indian Ocean. And so you can see that when you add viruses, you get a lot more ammonium in the system. And so what's happening here is, so we're in the process now of quantifying it. And so what we have is we've got bacteria out there which are susceptible to viral infection. And so the viruses come along, they attach to the bacteria cells, they lyse it, and so they release this dissolved organic matter through the viral shunt. And so then that dissolved organic matter which is released, there's also lots of bacteria out there that are resistant to infection, or there's no viral pathogens at that time. They take up that DOM, they deaminate the nitrogen to get at the carbon, and they release the ammonium. Okay, so they're just, it's the pathway that we've always known about. It's just that viruses, it turns out, are incredibly major players in liberating the DOM, which ultimately is fueling that process of deamination by bacteria and the production of inorganic nutrients. And so this whole idea of the shunt then, the viral shunt it's not just ammonium, but it's phosphorus, it's iron, it's all of these nutrients that viruses turn out to be intimately involved in. So Steve Wilhelm, for example, his group has shown that he can, in certain areas, he can account for all the iron that's required for the primary producers as the result of viral lysis in certain areas, and that of course that organic iron is taken up much, much more readily when it's organically complex, for example, than inorganic iron would be. And so then we also have this other idea which I think needs a lot of people thinking about, which is this idea that viruses are actually changing the stoichiometry of the nutrient recycling that's going on in the surface waters. And the process the idea is pretty simple. You've got heterotrophic bacteria with a C to N ratio of 4 or 5 to 1, and you've got phytoplankton with maybe a C to N ratio of about 7 to 1 or something like that. And as they pass through viruses, they're lysed, and the result is that things like phosphorus and nitrogen and things like that are going to be much more labile, for example, than cell wall materials and things like that. So you'll actually end up skewing the C to N ratio and making it much higher potentially. And so the question is, does that process in fact affect things like the biological pump? Right? So is it changing so you could imagine the biological pump, the efficiency increasing. And so again, these are ideas, these are things that need to be tested. And so we also have this idea of what's called the microbial carbon pump now, which is where does all this dissolved organic material accumulate in the ocean that's recalcitrant. So you can imagine, again, viruses having a key role in that by lysing cells and essentially releasing highly labile material and other material which is much more recalcitrant and ultimately gets concentrated in the system. And then so the point is that viruses, there's lots that we don't know about it. So there's really fundamental and important questions that need to be addressed of how to incorporate these viral, not only to quantify, and also to figure out how to incorporate these viral mediated process into aquatic nutrient and energy cycles. Because they're actually changing the way that nutrients are mobilized and how energy is flowing through those systems. So there's lots of things, fundamental stuff that still needs to be done. And then I want to talk about biodiversity finally. And so this is the last thing I want to touch on. So if we think about biodiversity, I work in my lab is in a biodiversity research center. And so my colleagues, would sometimes will say to me, well, you don't work on biological diversity, you work on genetic diversity. And so, it seemed hard, because these are ecologists and evolutionary biologists that I highly respect. And so I said, well, what do we mean by biodiversity? And so I love this definition, which is the one that they use at the United Nations, saying that biodiversity is the totality of genes, species, and ecosystems in a region. And so you can argue pretty easily that if you have no genetic diversity, you have no biological diversity. If everything's a clone of each other, you have no biological diversity, you have no ecosystem diversity. So the only way that you build up species diversity is by having genetic diversity. And so ultimately, I love this depiction of a pyramid which really shows that you have this underlying genetic diversity, which leads to species diversity, which leads to ecosystem diversity. And essentially, no ecologist will argue then that it's the genetic diversity which is underpinning the biological diversity. So if we ask the question is, where is the genetic diversity in the planet? Where does it exist? The genetic diversity, I will argue, is certainly, we all know it's in the small fraction, but I would argue that most of it is actually in the viruses. And so a collaboration that we did a number of years ago with Forrest Rower's group, we have, one of the things we have in my lab is the technique that learned in Jed Furman's lab was to concentrate all these viruses from seawater, and we've been doing this for years and years. We have like 2,000 of these now from all over the place. And so we just concentrated viruses from all these different locations. And just in a summary graph, there's lots of interesting things that came out of that study. But the thing that was the most profound at the time is that more than 90% of the sequences that we obtained using high throughput sequencing, we could find no homologs in any database. That meant that 90% of those sequences had no obvious matches in any database. And so that means that those potentially are encoding for proteins of completely unknown function. If we look at RNA viruses, we see the same thing. Alex Cully, who was a PhD student in my lab, and Andrew Lang, who was a postdoc, Alex and Andrew, looked at RNA virus metagenomics, and we saw the same thing. We saw that most of the sequences that we pulled out had no homologs in any databases, and the ones that did have homologs were in a whole range of different families of RNA viruses. But most sequence fragments, again, remained unknown. Jessica Labonte, a PhD student in my lab now, who's just finishing up, purified single-stranded DNA from the oceans and looked at that. And again, 90% of the sequences that she pulled out had no hits to any other sequences in any database. And then the 10% that did have hits had hits to all the known families of single-stranded DNA viruses. And so how do we make sense of this kind of metagenomic data? Because I think we're all kind of wrestling with these sorts of things, and how do we tie it into ecological processes? So one of the things that we started looking at the single-stranded DNA viruses, well, 90% of it had no homologs to anything else, and so we couldn't align it with anything, basically. But what you can do is you can do frequency profiling, and in this case take heptamers, and by doing that you can actually recreate the phylogeny of all the known single-stranded DNA viruses using this frequency profiling. So we can come up with this, and so what that allowed us to do was also to cluster, to come up with all the clusters of genetically distinct groups of single-stranded DNA viruses. So there's 7 families of recognized single-stranded DNA viruses. At the end of Jessica's analysis, we ended up with 645 complete viral genomes in 125 evolutionary groups that had no homology to themselves or to anything else outside of what they were involved in. And so potentially we have 120 or so new families of single-stranded DNA viruses from just a few samples of seawater. So the diversity out there is absolutely astounding. The neat thing is that when you take that, you can then take all that metagenomic data and lie it over the reads that we'll have from those metagenomic data, and now we can place about more than 70% of the reads into one of those genetic groups that we've identified. So again, that's one way of trying to come to grips with it. But ultimately we're trying to get to ecological solutions and trying to figure out what's going on. Another way to try and do it is we've been cloning in fosmids. So these are vectors which will take about 40 kb of nucleic acids. And so you can clone in these. And so the key is that when we go, when you looked at that data that we collected with Forrest Rower, where we have 90% of the sequences, typically now it's about 70% or so, have no obvious homologs in any databases. They're orphans. So what can we do with that information? We've just got sequences. So the only way we're going to make sense of it, we have to put it in a genomic context. And so what we've been doing is cloning whole pieces of viral DNA into large fosmids, and we're just lucky that the average viral genome in the ocean is about 40 kb or so. So you can clone most of a viral genome into a fosmid, and then you can ask the question, what's similar to it? And so you can go out, in fact, and you can start to look at different metagenomic data sets, and you can start to map them against the different fosmids. And so you can start to find genomic homes for all of those orphans. And so once we have a genomic home for them, then we can ask what are structural genes, what are functional genes, what are they doing? And then the other thing that comes out of this is we can find things like homologs up here. So this is a perfect homolog, an identical homolog to a bacterium which was metagenomically sequenced, which turns out to be an incredibly important player in oxygen minimum zones. And so now, by finding a perfect homolog in a viral genome, we've now recognized that this virus must infect that bacterium. And so then you can start developing ways of trying to ask the question, how many of those bacteria are infected by how many of these viruses, so you can start to try and build the ecology. So ultimately, we can sequence collect all we want, but unless we can start asking or addressing important questions, we're just stamp collecting. And so that's part of the challenge, I think, that everybody has to do. We've got to get some good minds out there to try and think, how can we put this data together and try and address important questions? So the point here is most of the genetic diversity and hence biologically diversity lies within the viruses, and the challenge then is to understand the functional significance of that diversity. And then the final point I want to talk about is we can also pull viruses out of the ocean, and that's absolutely critical. There was a great paper that came out last week in, I think it was Science, out of Steve Giovannoni's lab, in which they isolated a bunch of viruses which infect one of the most abundant bacteria in the world's oceans. And it's completely unlike other viruses that we've seen before. And so if we're going to build the ecology, we also need systems. And so one of the viruses that we isolated a long time ago in Texas infects a little microzooplankton called Cafeteria rowenbergensis. And it turned out to be a giant virus. And Matthias Fischer, a PhD student who finished in my lab, this is one of the most complex viruses that's known. And when we looked at this, it was intriguing because you see about half of the sequences, half of the coding sequences have no homologs to anything in any databases. And then the other half have homologs, the closest homologs to essentially all the domains of life. Okay, so this virus is essentially mixing up all the genetic information from eukaryotes, bacteria, archaea, other large viruses, and other small viruses, and they're all on the same genome. And so you can imagine the evolutionary significance of something like that. So, and then the other little piece that came out of this, which was so fascinating, is that when we were looking at it, we kept on getting a sequence of another virus, like a whole genome sequence that we thought must be a contaminant. But when we looked at the electron microscopy, we saw these little virus particles in there. And so you see the big virus particles, this is the Cafeteria rowenbergensis virus, and then these little viruses would multiply around the outside of the big viruses where they were produced. And so here you go, this little guy we called Mavirus, and the big one we called Cafeteria rowenbergensis virus. The amazing thing is what happens, the cafeteria, the microzooplankton gets infected by a big virus and then goes out and transports in the little virus from the environment. The little virus replicates on the big virus, killing it, and then rescuing the flagellate so it can survive. So it's like an immune system for viral infection. And so when we sequenced the little virus and asked the question, what is the little virus related to? It wasn't related to other viruses. It was related to large transposable elements that are found in eukaryotes. And some slime molds, 30% of the genome are made up of relatives of this little virus. And so at least the hypothesis that we put forward is that these little viruses were actually incorporated into the genomic information of eukaryotes to give them immunity to infection by big viruses. And those big viruses now are long extinct. And so we have all these large transposable elements that are just jumping around. And so there's so many things that we can discover by looking at that diversity that's out there. The Economist picked up on this story, which I had absolutely no idea about, and this was during the Nagoya Convention in Japan on biodiversity. And it was just a fantastic article because I wish I could have written something so elegant. But he recognized, the person that wrote this, he just said that biodiversity is not about tigers and whales and butterflies or coral reefs and tuna and these things. It's about all these little things that are too small to see, that are too abundant to count, that ultimately whereas most of that biodiversity on Earth resides. And with that kind of closing message, something to kind of take away with it. I have one of my graduate students, Caroline Chenard, works at 83 degrees north up here on the Markham Ice Shelf, and this is a collaboration with Warwick Vincent, a super scientist who's also, I believe, getting the Margalef Award later on this week. And you probably didn't know that, like, penguins are rampant in the Canadian Arctic, right? But polar bears will take care of them quickly, I'm sure. But the thing is, you go up there and you start looking at it, and so she works up in this far north, and what you can see here is down here is the Markham Ice Shelf, and so all along here you can see these ice shelves, and behind them are these big fjords. And what's remarkable is these ice shelves, the ice is extremely deep, and so they form these massive freshwater lakes behind them that are 30, 40, 50 kilometers long, and maybe, I don't know, 20 or 30 meters deep. And they've been freshwater lakes for 30,000 years. And if you look at the microbial life in those systems, they don't look anything like the other microbial life that we see in other systems. That makes sense, right? I mean, they're in a very, very unusual environment. And so you can see here, a year later, the Markham Ice Shelf is completely gone. And the result of that, so here we have August 6, 2008, August 12, 2008, that whole freshwater lake just drained and was completely replaced by seawater. And so we've lost this huge ecosystem without really ever knowing what was there. Now, we've sampled some of them, and we know that the microbial life there is completely different than the microbial life that we've seen elsewhere. And so the take-home message is that maybe we're just a little bit ahead of the curve here, but we need to be thinking, the more that we learn about the diversity, the incredible diversity in that microbial component of the biosphere, the more we realize just how little we know. When we talk about 80% of the coding sequences having no homologs in any databases, that means every one of those is coding for a protein that we have no idea what the function is. Right? And so we need really smart people to start thinking about how we're going to dissect this information and try and understand what the biological significance of it is. So the take-home message, I guess, at the end, and I hope I haven't gone too long, is challenge yourself and those around you. So don't be afraid. Don't be afraid of going out there and testing your students and encouraging them to be their best. Your success depends on their success depends on your success. And so you really are in a symbiotic relationship. For those of you that are interested in these kind of questions, think about ways to try and incorporate viruses into these biological and geochemical processes. It's a wide open area, and there are some smart people that are trying to do this now, but we need a lot more people that are smart. Biological diversity resides in genetic diversity, and if we think about all of us, we're working on, probably just about everybody here is working on some microbial component, and I'm including all those eukaryotic phytoplankton, those protistan grazers, and everything else. A large percentage of us are working on that component, and it's just so incredibly important. And so we need to do our best to go out there and educate people and let them know that it's responsible. Every other breath we take is the result of oxygen that's produced by this microbial life. And ultimately protecting this biological diversity means protecting the microbial diversity. So I have to thank, this is my cohort of students and postdocs in the lab, and again, I owe my gratitude to all of them and all the colleagues. So thanks very much.